project that involves environmental education and also creating science literacy coupled with art. And so that's what this workshop is about. I also want to encourage you to consider, we have a sustainer campaign on now. So as the project coordinator for Nature Lab, I would love it if we could get some sustainers tonight. You could donate $1 a month. A sustainer keeps you, giving money. You all know that concept, right? And so it'd be really great if anybody wants to sign up. There's ways to do it either online or in the back. We can facilitate that. And just this place is so awesome, and there are very few places in the world like it. So we just are trying to keep it going and want you all to participate in any way you can. So thank you, and consider that. So tonight, I'm really excited to introduce a dear friend of mine and a colleague who I have admired for many, many years. Her name is Marta De Menezes, and she comes here from Lisbon, Portugal, although she was actually in Toronto doing this workshop, and Windsor, Canada, doing this workshop previously. She is an internationally recognized artist who has had shows all over the world of her artwork. She started making work in the 1990s-ish, at least 20 years ago, that dealt with biological materials in her artwork. So she's going to be talking to you about that in her talk tonight to introduce the workshop. Marta has just been a real force behind the whole movement of what we call bioart, which is a very kind of big umbrella for a lot of work that a lot of us, including myself and others, are doing that involve working with living systems with art also. And so I think that you'll see that her work has a kind of real human resonance and a profound effect on people who see it, and I hope you feel the same way I do. So without further ado, please welcome Marta De Menezes to our team.
So is this working? Yeah. So thank you for coming. Thank you, Kathy, for inviting me here. Thank you to everybody at the sanctuary for all the help setting up and making this a reality. Just to give you an idea, so Kathy made a great introduction of what I do. I think, so first, I'm giving you an artist talk. I'm not specifically talking about or explaining what CRISPR is with this talk right now. And I won't do this today, even for the ones that are going to join the CRISPR workshop. There's many different reasons for that, and I will go on explaining those at the end of my talk. But for now, this is an artist talk, so I will be talking about my art. And it's really important for you to understand. I thought every time I do an artist talk, I think it's really interesting. I hope it's interesting for people to understand how I go about making my projects. So I put this slide on. This is not a slide that I made, obviously. This is design thinking slide. So it's a diagram that I took from another field of knowledge, and I did it on purpose because as an artist, I had diagrams like this before in my talks, but I never dared put empathy or empathize in the first square. And when I saw this, I thought, okay, of course, the driving force behind all of my projects, behind probably all of anybody's projects, is empathy. It's the love for whatever you're doing. It's your curiosity for whatever you're interested in. And that is what drives you the most. This is what makes you then choose to pursue a specific thing instead of another. So it was really interesting for me to realize that this should be a recognized step into a methodology, whatever the field that you're working. And so I start by liking what I do. And the drive is that I ask a question or that I find a subject that really I feel passionate about. And so throughout my 20 years doing art in biology, I found that there's an umbrella theme that I really, really like, and that most of the works that I can think of, most of the questions that my mind comes up with are related to identity. and maybe I never overcome my teenage years trying to find out who I was, but I still really do believe that knowing yourself is probably the best weapon I have to interact with the rest of the world. Maybe not weapon, maybe just the tool, maybe just a method, maybe just a way of engaging an interface, but knowing who I am and what I am will help me interact with everything else. So all of my works start with a question, or start with an idea, or start with a curiosity about a specific project or a specific, maybe it's a technique, maybe it's a material, maybe it's just something that I come across that that changes my perspective on identity. So I'm a strong believer that identity is not a stable thing. It's not a static thing. I'm always changing. I will be someone else after I talk to any of you. So this means that whatever project that I do, I need to learn a different technology, and more often than not, I'm using a different material. And in terms of live material, if you're working on identity, I think it's really important to understand the identity of whatever you're working with. So it's sort of like a reflecting mirror, if you think. If I'm working on a different organism, I am sort of mirroring my understanding of identity and about myself into that organism. This means that I've started working in an evolutionary biology lab, modifying butterflies, and questioning what is a natural organism, and if you change it, but you don't really change it, then is it still natural or not? And what are the criteria that you're using to determine something like this? I then moved on to do many other things, including a protein that has my name as its amino acid sequence, or a tree that is made out of neurons, and it's actually shaped like a live neuron. I've created Unbiased with a friend of mine that I met specifically to make this piece, who is a data visualizer. He's an expert on this, and we worked together on 4 projects, 2 digital ones and 2 biological ones. And the question that I was posing is, can you actually feed into a system data and have that system, that live organism, or that life ecosystem actually express the data that you're feeding to it? And that was a really interesting challenge. I've worked in microbiology labs. I have worked with human cells, with HeLa cells, for those who know what HeLa cells are. I've worked with functional magnetic resonance scans, cancerous cells and immortal cells, blasts, so some software, some comparison DNA. And lately, I've been interested in CRISPR, and actually my interest in CRISPR started from the first project that I did, the butterfly project. Okay, so I will talk about 2 of these projects that I'm doing with CRISPR, and that will be the end of my presentation today. So just before I go into CRISPR, I would like to talk about my most recent projects. And one is maybe not that, my most recent finished projects. Maybe I should add that little word there. This project is called Immortality for 2. I'm very proud of it. And not only have I been working in biology labs for 20 years, I've also been married to the same guy for 20 years. And he's an immunologist. Okay, so actually this work didn't start as a collaboration with Louise, my partner. It started as my interest to work out the meaning of immortality. What does it mean as a word, as a concept, and how is it used in different fields of knowledge? So the thing that triggered this, actually the 2 things that triggered this, but they all came from the same book The Immortal Life of Henrietta Lacks, the HeLa-cell line source. I'm not sure if I'm not very comfortable with these words around that, but I haven't found better words yet. And so, the book is amazing. It's actually a really interesting book to read. I had worked with HeLa cells before, and just this idea that you have a very mortal disease, but the name that you give to a line of cells is an immortal cell line, just got me wondering, what is this? How do you understand immortality this way? What does it mean to be cancer and still be called an immortal cell line? And so what is this immortality, and how does it relate to our understanding of immortality if you're not talking about science at all? So I came to the idea that, can I make my own cells immortal? How is that going to allow me to think about this? And I cannot. So technically, it is possible to make any cell into an immortal cell line. So nowadays, you don't have to get primary sources from cancer patients, but you need to insert oncogenes into that cell, and that is a health risk. So obviously, I'm not going to go that way. So I was stopped very early in this process of what does it mean to be immortal in this way? What does immortality mean? To create a line of cells that are yours, outside of your body, that are actually cancerous cells. So how does that work? And so at the same time as this was a process, I was thinking about immortality in general, and how we conceive of immortality as humans, and how immortality for us is always attached to other, to non-human. So first immortal beings are probably gods, if you think about it. Whatever god you believe in, they're immortal, or they attempt to be immortal. I don't know any mortal god. Or superheroes, vampires. There's lots of others who are immortal, and part of the immortality, or gaining immortality, is the loss of humanity some way. So that for me was really interesting. At the same time, who wants to be immortal alone? Not just who wants to live forever, but who wants to do that alone? And so this was the point that I came to my partner, Luis, and asked, would you consider this journey with me? Would this be an interesting project for us to do together? Fortunately, he's very open-minded about these things, and he automatically said, yes, sure, but we need to do it with with immunology cell lines, so immune system cell lines. Which, okay, we're achieving immortality together. Can you please? And we did that. And so the health and safety issue was not an issue anymore, because I could transform Luis's cells into immortal cell lines, and he could do the same for mine. And we did it in parallel in the lab, in his lab. And it was white blood cells, so it was not very difficult to access the cells that we were going to use. But they are immune cell lines. So what happens when you put them together? They kill each other. So the price, again, there was a price, after all, which was perpetual isolation, please. And this is how you see it. So actually, the installation piece has living cells. There are cells, there are immortalized cells, but they are opposite sides of a very big table. Not only are they, so they're there, and they're cancer cells, and so they're very, very prolific. So they actually don't need an incubator to survive. They can survive and proliferate at around 25, 26 degrees Celsius, which I don't know what it means in terms of Fahrenheit, I'm sorry. And actually, the only place where these two cell lines can be in the same place together is in the middle of the table on the overlapping area of the two projections. So under each flask, you have a microscope that is attached to a computer, so they're both attached to one computer, and the computer is attached to two projectors. So you see the live cells being projected onto the table, and there's an overlapping area, and this is the only place where Louise and I can be together through that kind of immortality. Of course, we have 2 kids, yes? So that is more or less sorted. But again, part of it is about questioning what is this? Is it achievable in what way? And what is the price of achieving something like this? So that was the first project that Louise and I did together. And we did this in 2014. So quite a few years after we actually got married. Then in 2017, we both went to a lecture by this gentleman over here, John van Rood. And John van Rood is the father of clinical transplantation. So actually, Louise started his career as a PhD student doing transplantation. He did it in Oxford. And so this was one of his heroes. The talk was really interesting for me too. He was a wonderful speaker. He died in 2017, so a few months after we actually saw him speak. And he was talking about experiments that he did in the 60s that gave rise to the whole field of clinical transplantation and to the description and recognition of HLAs, which are human leukocyte antigens, that help histocompatibility tests and therefore help matching donors or organs to recipients. And this is why we have clinical transplantation today. So the experiments that he did, if you could show the next slide, gave rise to some very, very important papers in the field. And he was describing those experiments. So the experiments were these. So amongst his colleagues, they started asking for volunteers to do small skin transplants. And this is what you see here. And I don't have the damn thing. So I'm going to have to move. So you have a self-transplant here, and then you have the transplant of 4 different donors there. And this was done amongst colleagues in the lab, and then colleagues in the institute. And of course, nowadays this would not be possible or approved, but it happened, and actually this is why we have clinical transplantation today. So when I heard about these experiments, I was mesmerized. This is amazing. And living with an immunologist for 20 years, you sort of hear the 2 words self and non-self repeatedly. And working in the umbrella subject of identity, self and non-self are like, okay, I want to do something with this. I just never really realized what I wanted to do until I heard this man talk. And this often happens to me. So, self and non-self are really interesting concepts. The immune system is so complicated that I always felt a little bit intimidated in trying to go into, do I really want to know that much about immunology to be able to come up with a project specifically on this? And so when I heard these experiments, this was the click moment. Okay, this is something that I would like to work on in terms of thinking about self and non-self. And I'm not sure if it was Louise who proposed the idea and I said yes, or if it was the other way around. So he will tell you, I came up with the idea and he said yes. I will always tell you that it was him that started the idea and I said yes. It doesn't matter. We both said yes. And so the proposition was, let's make a skin transplant between each other. And let's reject each other at a molecular level. And this is what we did. Okay, so for me, again, And this is in no way me trying to explain to you what a relationship should be like. I'm not giving any kind of relationship advice. But for me, living with him, can you go back? Living with someone for 20 years means periodically that I have to reaffirm myself as self. I am me, there's no such thing as us, and you go away. Not necessarily go away, but I do need to remember who I am, or to restate who I am, or to reassess who I am at any given time. And sometimes it gets confusing when you're living with the same person every day. And so for me, it's really interesting because this project is very much about this. You know, there's no more intimate way that I can reaffirm that I am myself and not him or not us than to have my body physically reject a piece of his skin.
Through my experience in scientific laboratories, I actually see no difference in terms of the amount of creativity that the scientists need to have to develop their projects.
For me, the most important similarity is that both of them rely on a concept, on an idea that needs to be exploited until it is proven correct in the case of science, or until it leads to a developed artwork in the case of art.
My new project, Marta, is actually in a way a follow-up of Immortality for Two, and it consists of inducing our bodies, Luis's body and my body, to produce antibodies against each other.
Marta starts with an idea that recapitulates very important experiments that were critical for the development of transplantation as a useful procedure.
We're going to reproduce a famous set of experiments that led to some important discoveries in science.
Van Rood in the Netherlands discovered the rules of histocompatibility to get a donor and a recipient matched.
The surgeon is going to perform the surgery, and they're going to remove a little portion of skin from me and Luis, and they're going to exchange that.
And what will happen is that a surgeon will remove these small bits of skin from my arm and Marta's arm, and will make a transplant.
And the process of rejection will start then.
So what Van Rood realized is that within our genome there are proteins that are really important in presenting bits of viruses and bacteria that infect us to the immune system. Of course, that between Marta and I, we will have many differences in these proteins. So it is inevitable that the cells from the immune system will be activated by these proteins, and as a consequence, rejection will happen.
Marta is, for me, a really interesting challenge. Very much in the same way that Immortality for Two, there's an impossible endeavor of being together in the same place as Immortal. Marta has this impossibility as well of becoming one.
It is also a continuation of the exploitation of how two people that love each other can share a pact.
You notice, maybe you didn't, or what my point is, so one, I wanted to show you a little clip of the video because it's always important to me that I'm not speaking for Luis. This is a collaborative project. And so having you hear the way he explains the project is very important for me. But also, you can see that for him, it is more of a pact. I don't know if you noticed that. So this pact is actually a life commitment. So if you know about immunology, these antibodies that are produced as our bodies are rejecting each other are actually a little bit like a vaccine, but also you can see it more of as a memory aid. So we will have forever molecules within our body that will forever recognize each other. And this is the commitment part. It's like there's this memory aid that we will never stop recognizing each other. Of course, we will reject everything, but but it depends on your perspective. And actually, immunology is trying very hard, at least the immunologists I know, to get away from this warfare kind of understanding of what an immune system is. And I think that for Luis, this is very much part of his interest in doing this, to try and see how the immune system can be thought of as not just a fight against, but also a memory or a remembering of things. The piece is shown with the video. And it's also, of course, I'm not going to do the experiment every time. I'm not going to do a transplant every time. This is the scar of in my arm, and it's, you know, Luis will tell you it's textbook because it worked really, really well. So you have a self-transplant here, and this is where Luis's skin was, where I rejected him, and now there's a scar. And because I wanted people to understand the process and to understand the rejection and to understand the commitment as well, we came up with the idea that we could have the projection of the surgery happening in the gallery space. And actually the screens for seeing the transplantation happening were the arms of the people from the audience. So if you go to the exhibition space, you have a table with chairs on each side, so you're facing each other, and you can put your arm on the table and you'll see the transplantation happening, the surgery happening on your own arm. And that will give you a much better, or much closer feel of what it is to commit in this way. So these are my two most recent projects. I can now move on to talk a little bit about CRISPR and why I decided to pursue that avenue. So nothing in biology makes sense except in the light of evolution. I'm not so sure. Okay, so, and I'm not the only one. And this is one of the things that I've learned working with biologists for about 20 years. And so especially, and in this case, you can see it was in '64, it would have been mentioned as Darwinian evolution. So the idea of evolution has changed very much over the last few years. So the DNA, not the discovery, so the description of DNA as a double helix happened in 4. And so since then it has changed a lot. Can we go to the next slide? And thinking about CRISPR, so I don't know if you know so I did mention before that I did these butterflies, or that I altered these butterflies and asked the question, were these butterflies natural or not? So when I did my first project in an evolutionary biology lab, the technology that I was using was not genetic, and it was on purpose. Okay, so it was a very conscious choice to alter the wing patterns of live butterflies without using genetic manipulation. It was also the methodology that they were using in the lab. And the thing that really made me want to go for the non-genetic was that each butterfly was unique. So every butterfly that I altered was in itself an art piece, in itself a question. And the thing was, for every butterfly that I modified, there was no difference from that butterfly to the wild type. So the offspring of that butterfly was back to wild type. There was no scars on the wings of the butterfly. There was no lifespan difference. There was no mating behavior difference. This was all part of what the scientists were researching in the lab. So it's not that I found those things, but this was part of the research that was done in the lab, so I knew the answers to these questions. And so, what makes that butterfly man-made except for the fact that it is asymmetrical? And that would not be possible if it was done genetically, okay? So the question was, are these natural butterflies or not? And for that specific question, it was really important that I could use a live material that was not transformed through genetic manipulation. When CRISPR came about, it was really interesting for me to think about how it was not conceptually a very different idea from other ways of genetically manipulating other organisms, or organisms in general. We can pretty much do a lot of things to organisms through the genetic line. And they're pretty, well, they're not very efficient, but you can still do it. The thing that really struck me as interesting was the breakthrough was efficiency and cost. And the impact that those two little characteristics had in the sciences were outrageous, as you probably have heard. So they were so big that between 2011, which was when the first CRISPR papers came out, to 2015, the amount of funding that projects like that got just went through the roof. And of course, as some of you may have heard, we now have two babies that have been genetically modified through CRISPR in China. So, but before all of that, or before the babies, I started being interested in CRISPR because it allowed me to pose some questions, and a lot of the issues being raised with the use or misuse of CRISPR were things like, are we going to create, what are we going to create with CRISPR? Where are we going to go? And it's always forward, right? And I was going, can we go backwards? Is it interesting? Does it make sense? And how does it work? And so my idea was to propose an origin of species kind of concept. Can we find out what was there before? Can we go backwards in time using CRISPR? Does it make sense? And I've learned a lot since asking this question. So my first project that I can talk about today is actually a project where me, an artist, and we can call ourselves a different species, maybe, a philosopher and a scientist, are trying to develop a project where all three fields of knowledge develop a relationship to create a new organism, okay? And the motivations for creating this new organism or old organism is different because we come from different fields. So for me as an artist, again, it's an issue of identity. And in this case, the material, and not the technology, determined what kind of perspective on identity I wanted to pursue. So actually, I'm working with a philosopher who's Maria Antonia, she's from Mexico, and a scientist who's from Portugal. He's also called Luis, but he's not my Luis. And he's also an immunologist. But he doesn't work with organisms that have an immune system. He works with Drosophila fly. And he works with, his main interest is the interaction between host, Drosophila, and a pathogen, which is the Wolbachia, which is actually a symbiont relationship with the Drosophila, which is interesting. It's a pathogen, but it's also a symbiont. Basically, the Wolbachia bacteria, which they cannot grow outside of the Drosophila fly, so you cannot take a petri dish and grow it there, or take a little, whatever, LB medium and grow it there. The pathway of the interaction between these two organisms is so intricately connected that having Wolbachia in their cells increases their lifespan to up to 3 times as much. Okay? Because it gives them resistance to viruses. So this is interesting for them. For me, it's a way of thinking of a joint identity, a composite identity, an identity of 2 organisms or 2 species that is with difficulty separated, or that it actually transforms the identity in a way of the fly if they don't have the Wolbachia in them. So for me, this is the aspect of this. The project itself became a project of the phylogenetic tree of the Wolbachia and the Drosophila fly, and these are the images that you see there. So when I say 3 fields or 3 species coming together to think about this, we have, let me try to explain a little bit of this graphic for you. So the green line that you see there is actually the cell wall. The green line that you see here on this corner, the left bottom corner, is the nuclei of the cell. Every little shape that you see there is 1 gene, and they're all named. And the label over there just says that, so if you have a red gene, it has a very high ratio of mutation. If you have a blue gene, it has a very low ratio of mutation. And so all of those arrows that you see pointing into the nuclei are actually pathways of interaction between the Drosophila and the Wolbachia. So you have the JAK-STAT pathway, you have the TOL pathway, you have the IMD pathway, and maybe that is a secondary one. And in each of those pathways, you have the genes which are involved in that interaction, and they're studying one of those. So they're studying this one in the lab. So first thing that you need to know about CRISPR is really difficult to change the whole thing. What we needed to do was to devise a strategy. How are we going to alter this gene so that the objectives of all 3 of us are interesting? Okay, so I haven't talked about the philosophy yet. Can you move 1 slide further? So if you have a gene, and I'm sorry, the resolution is not brilliant. It looks great on my computer, by the way. What you have there is a list. So on the left side, you have a list of not only the Drosophila, so the ones that start with a D here, above this line, those are all Drosophilas. But then you have other insects, or mosquitoes, actually. And you have in front of them the sequence of amino acids that gene specifically produces. And basically what you do when you try to figure out what was there before is you compare them and see if the amino acids are different. And when they're different, you go, okay, I wonder which one was there first. You go into the phylogenetic tree and you try to figure it out. But again, so this is the first thing, is this is an educated guess. The first thing that I learned about this is in a phylogenetic tree, you only have the edge of the tree. You don't have the branches before. So there's no way to compare. So basically, you're making a very good educated guess, nothing more. So when they tell you that they're going to de-extinctify, I don't know how to say this, to bring back any organism, you know that, well, they're making one up. As close as they can possibly get to what was there, for sure. But it's still, there's no way of actually finding out if that was there before, besides the fact that at least half of it is going to be, so if you do a woolly mammoth, you need to have a womb to grow it in. And that's going to be an elephant womb. And that's going to be the mummy. So a lot of other things are going to be involved in creating this organism that is not just the genetic code. So it was really interesting for me, in physics, and I do I do love physics. It's impossible, it's impossible to go forward in time, theoretically, but maybe not go backwards in time. In this case, in biology, it seems to be completely impossible to go backwards in time. We just don't have enough data. So this is really, really interesting for me. I'm sorry, just next one. So here we go again. What are we going to do through that gene? And for me, and for Maria Antonia, it's going to be about trying to find out what was there before. For the scientists, it's really about, if you change what you have there, how is that going to affect the interaction between the two organisms? And actually, this is, for me, a really interesting collaborative effort in the sense that that completely works for all three of us. So for Maria Antonia, and I ask you to read it, because again, I am not the best person to speak for her. She, as a philosopher of nature, this is her interest in the project. Okay, so thank you.
So as a non-scientist, the idea of scientific immortality is something that I need a little bit more information about. What makes a cell line immortal? I haven't read the Henrietta Lacks book. I should.
Yes, you should. But just for fun.
But what is it that just simply, why don't cells just die?
Well, they do. So if a healthy cell will die after a certain number of divisions, and an unhealthy cell will not. And so that internal clock that tells them to stop dividing at a certain number of divisions and just die is not working. And this is actually one of the definitions of cancer. And those genes are not functioning properly, and so it just never stops dividing, and this is why they call it an immortal cell line. So it's very perishable, because if you have mammal cells, whether they're cancerous or not, outside of the body, open air, they're going to die from, just pathogen infection or just, but they're resilient, and if you keep them sterile, they will just indefinitely divide.
You tangentially mentioned the, I think, Stanford trained Chinese scientist who went back to China and tried to make
Babies.
The babies of one or two of the parents were HIV positive, and create children that would be immune.
Yes.
And also, I think they discovered that the two children, not only are they immune, but they're more brilliant than they might have otherwise been. But that scientist has gotten into a great deal of, you're messing with God problems with the regulatory agencies.
Mm-hmm.
And what you're doing is what you're doing outside of those harassments?
Well, what I'm doing is outside of the harassments, because I'm not working with humans. Definitely. What I do is so whatever I do with CRISPR, I'm doing it within a lab that has to obey the rules. So, and they do have to face ethical boards. So they're not going to challenge those ethical guidelines, because they don't want to lose their funding, basically. But in terms of flies or bacteria, the rules are not very complicated. It's when you get to not only to human manipulation, but to germline human manipulation. So a lot of the HIV research that is being done to try and, it's not actually to try and immunize against HIV, it's to to actually try and develop therapies that will allow an HIV positive person to reduce the amount of cells in their body that are HIV positive. And because CRISPR has that characteristic that not only can you manipulate a germline, but you can also manipulate individual cells, and you can do that in an adult organism. Most of the CRISPR that's being done all over the world is being tried in plants, which is not very successful, because even though they do accept the cutting, the ratio of acceptance of a template that we give them is not very high. So they, I think, pretty much gotten over the idea that they could actually induce a plant to take a code in through CRISPR. So they still go back to the old, not very old, but more traditional way of manipulating the genes. A lot of mammals, yes, but again, it's more about trying to do therapies. The first genetic manipulation summit that was in Washington in 2015, 16, that you were there. 15? You were there. Had a public statement written down afterwards that was very cautionary. And the last one, which was just after the babies were announced, which was in Hong Kong, if you see the public statement, it is an after the fact kind of statement where it's not about should we do it, is now that it's done, how do you proceed? So again, I would be very skeptical that the babies are smarter because of that specific genetic manipulation. How do you know? I don't think that you can know that it was specifically about that. They could just be really smart because they had very good nutrients and a very well monitored pregnancy. It doesn't the two don't relate. So if you see a news like that, please go look at the sources and see if the sources are in any way scientifically based, or if it's just media taking over the attractive parts for media, which are more often than not the scary parts. So I know that they did those the babies exist. I know they have been genetically manipulated. That has been very clear. The thing is, it feels to most of the scientific community as a proof of concept and not a necessarily specific useful manipulation, in the sense that you're not going to test if those babies are actually immune to HIV, because you're not going to infect them with HIV, right? So, that specific manipulation doesn't seem to be very useful. It just seems to be as an attempt to make a proof of concept that it can be done, and an opening of the field in terms of this is a given fact. We then need to think about what to do with a given fact.
What is it when an artist changes a gene, and similar to you having a scar on your arm, making a scar in a genome for a signature event? Is that, and I'm thinking about these babies, are they scarred by the manipulation, or is there something different about humans that makes it an extra scar?
I think you're just assuming that people understand what you mean by scar.
Like a remnant, like even the effect of the symbiont.
Everything changes you, right? You understand this. So every day you encounter a million pathogens that will leave marks in you, because you are going to be, your immune system is going to react. So you will have lots of molecules inside your body that will be just about acknowledging that in any given day you're encountering, I don't know how many different organisms. Yeah. Is that considered a scar? If that is considered a scar, I think you're scarred anyway, because your DNA is expressed differently depending on your environment. It's always So those scientists probably just didn't understood what you meant by scarring, I would say.
Thank you so much, Marta De Menezes. Let's give her a big round of applause.